I still mean it, that everything that you need to know about DNA structure is actually on this slide. So I'm going to run through it again at peril of boring you because we have done this before. Nonetheless, it's so important that you understand the structure of DNA in order to understand its function, because of course the two are intimately connected. So the very first thing we'll just discuss in general, nucleic acids, not just DNA, but all of the nucleic acids. There are two nucleic acids that are of particular concern to us, and those are DNA and RNA. And we need to understand, first of all, the difference between the two, but then also the import for their overall structure. So let's start here. This is the monomer. This is a basic monomer of which all nucleic acids are made up. It is a nucleotide. And a nucleotide consists of three parts. It consists of a sugar in the center here, a nitrogenous base. Nitrogenous base, that simply means a ring structure, very rich in nitrogen, which is slightly basic. It's only slightly basic, but nonetheless, it is classed as a base. And then on this side, here, the third part of a nucleotide is a or a set of phosphate groups. So there's this phosphate-rich area here, sugar here, nitrogen-rich basic area here. So those are the three units. You'll see this is a nucleotide. You'll sometimes see reference to a nucleoside. Nucleoside is simply this sugar part and then the base joined without the phosphates present. That would be a nucleoside. Most of the time we are going to be talking about nucleotides. And so let's revise each of these parts of the nucleotide because each are really, really important. First of all, the sugar here in the nucleotides is a pentose sugar. It consists of 5 carbons. And here they are. They're not all written into the structure here, but you can see that they are here. One there, one there, one there, one there, and then one out here on this branch here, there. So these 4 here, together with an oxygen, make up a ring structure. So there is this ring of carbon and oxygen with this carbon attached to it. We number these carbons. This one is the 1' carbon. So this is 1' carbon, 2' carbon, 3' carbon, 4' carbon, and then here's the 5' carbon. Those prime numbers must feature in your thinking all the time because they are significant. What happens at these different carbons is intimately tied to the structure and function of DNA, of the nucleic acid, whatever nucleic acid we're talking about. So here's the 1' carbon. At the 1' carbon, that is where this nitrogenous base is attached. This nitrogenous base may be 1 or 2 rings, depending on what it is. So there's 1 or 2 rings, nitrogenous base, attached at the 1' carbon. In all of the nucleic acids, there are only 5 of those bases that are used. And they are cytosine, thymine, uracil, adenine, and guanine. Cytosine, thymine, uracil, adenine, and guanine. Any of them could be attached at the 1' carbon. At the 2' carbon, depending on what nucleic acid is we're talking about, there is either an OH hydroxyl group or just an H group. And then wait a minute, if I've got an OH group and I take away an O, what have I done to it? I've taken away an oxygen and deoxyed it, okay? Keep that in mind. So this 2' here, there's either just a hydrogen or an hydroxyl group. The 3' here in a free nucleotide, the 3' here has a hydroxyl group. And I told you before, you need to go to bed at night saying the 3' hydroxyl, 3' hydroxyl, 3' hydroxyl. Why? Because the 3' hydroxyl on this nucleotide is the only reactive site. If ever I want to do anything with a nucleotide, if I want to join another nucleotide to start building a chain of nucleotides, I have to come in here and add it. I can't add it anywhere else. I can only add it at the 3' hydroxyl. 4' with nothing significant we need to worry about, excepting that it is attached to the 5' carbon. And here's the 5' carbon. 5' carbon has phosphate groups attached to it. And we'll see the significance of that in a minute. But in free nucleotides, often there are 3 phosphates attached here. So just think back and you'll realize that you've seen something that bears a strong resemblance to this when we looked at ATP, right? ATP is also a nucleotide. And ATP has 3 phosphates attached to it. We also saw ADP, which had 2 phosphates. And ATP and ADP, they also have obviously, they have a nitrogenous group, which is adenine attached there. Find adenine here, and you'll see it's a 2-ringed structure. Okay, so that's the basic structure of all nucleotides. But we have 2 kinds of nucleic acid. We have DNA, which is deoxyribonucleic acid, and we have RNA, which is ribonucleic acid. So what is the difference between the two? There are several. The first place you look for a difference is here at this sugar. This is a ribose sugar. That is basically a ribose. So in RNA, there it is there. That's the true ribose sugar. And it has an OH group in RNA. If I take away this oxygen, I create a deoxyribose sugar. Still a ribose, but it's a deoxyribose sugar. Here it is here, deoxyribose, lost that oxygen. This ribose sugar is what occurs in RNA. This deoxyribose is what occurs in DNA. That's the first difference. The second difference between RNA and DNA is that in DNA, we have this base, thymine. But in RNA, we use this base here. Very, very similar indeed, but this is uracil. So this is characteristic of RNA, is uracil. Thymine, characteristic of DNA. And both of these things, this difference in the bases and the difference in the sugar structure, in the pentose sugar structure, are labeling techniques for the cell. This is one of the ways the cell knows what it is dealing with, whether it's dealing with DNA or whether it's dealing with RNA, because otherwise they look very, very similar to one another. But these are ways in which the cell recognizes what it is dealing with. So for all of our nucleic acids, we have this basic structure. We have a nucleotide consisting of a sugar, pentose sugar, ribose in RNA, deoxyribose in DNA, always with phosphate groups attached here if it's a nucleotide, and with nitrogenous bases attached there. So this is our monomer. The nucleic acids are polymers, and they are often very, very long polymers of nucleotides. But you'll realize now that there in DNA and in RNA, there are only 4 nucleotides with different bases attached. Okay, so we can have, here's our basic nucleotide structure, sugar and phosphate. In DNA, there are 4 possible bases that could be attached here, and they are cytosine, thymine, adenine, and guanine. And in RNA, the same thing. We've got the sugar here and a phosphate. There are 4 possible bases. They are adenine, guanine, cytosine, and uracil in RNA. There is another difference, and it's a major difference. And that is that when we look at the structure of the polymer of the nucleic acid, DNA is a double molecule. We'll see how it becomes a double molecule in a minute, how that structure is created. But DNA is a double molecule, is a double helix. But RNA is not. RNA is fundamentally a single-stranded molecule. And I say fundamentally because you'll see that there's a little bit of a complication. But nonetheless, RNA is fundamentally a single-stranded, DNA is double-stranded. Okay, so let's just have a look and see how these nucleotides now get joined together, get polymerized into the long molecules that comprise the nucleic acid themselves. I told you, the only place I can do anything, the only reactive site I have on the nucleotide is here. It's at the 3' carbon. And if I want to make a nucleic acid chain, what I have to do is I have to bring in a new nucleotide here, and I'll react the phosphate of my new nucleotide with the hydroxyl group here. This is the reactive site on this molecule. And I'll bring in another nucleotide here, and I'll react it, and I'll join it by forming a join between the sugar and the phosphate. Okay, so what we look, when we look here at this simple chain of nucleotides all joined together, we have a sugar, phosphate, sugar, phosphate, sugar, phosphate. This here is a backbone, and it's an extremely strong covalently bonded backbone. And that is the sugar phosphate backbone to the nucleic acid, whether you're talking about DNA or whether you're talking about RNA. So that's the first thing. Here the backbone is, but this means that, remember, this here is 3', that is the 5' there. This here is the 1', and this is where these nitrogenous bases are. So they're pointing away from this backbone here. So here's something else to understand. In this diagram here, we have squashed our molecules flat just for our convenience so we can see it. But in real life, each time one of these bonds is formed, it doesn't form flat at 180 degrees. It forms in space, and it forms at a slight angle. So these all begin to add up in space. And what they do is they cast this molecule into a basic helical shape in space. So that's where we start to derive this helical shape to DNA, for example, is because of these slight angles that are subtended by each of these joins. So the other thing to point out is that I can join, let's just think about DNA for a second. Okay, we've got 4 bases: cytosine, thymine, adenine, and guanine. I can join them in any order. Just because I've got adenine here doesn't mean to say I have to have any, just because I've got adenine here doesn't mean to say I have to have anything particular here. If I've got guanine here, I can add any of the other bases. I could add another guanine at the end here. I could add adenine, thymine, etc. You can bring in any of the bases here. That's not to say that the order in which they are joined together is random. It is not. But the order is determined by genetics. This is the recipe that is lodged in the gene. The gene contains the recipe for the order in which these bases must be added. So the order or the sequence of bases is very strictly governed by the gene. And it is the sequence is something which has evolved. It has evolved over all of the eons, so that gene, the product of that gene, is operating always at optimum. This is the best that genetics can do. This is the best that the genetics can provide to the cell. Okay, so here we have a set of bases all joined together. They have different nitrogenous bases sticking away from the backbone. And in RNA, we have this backbone consists of ribose sugars and phosphates. In DNA, it is deoxyribose and phosphates. And the other difference we've already heard. In DNA, there is another strand. There is a strand which matches this on the other side. So the DNA molecule consists of two strands that run together with one another, helically in space, to produce this kind of screw-shaped shape in space. One backbone here and another backbone, as we'll see, on the other side here. And sticking across in the DNA, across the middle, are the bases. And in fact, the bases, the bases have, if I've got a base here, it has a matching base on the other side. And that matching base is there according to very strict rule. And that is that adenine always matches with thymine. If I have an adenine here, I must have thymine there. And if I've got a cytosine here, I must have a guanine here. If I've got a guanine here, I must have a cytosine there, right? If I've got a thymine here, I must have a uracil here. Just remember that, A to T and C to G. Adenine to thymine, cytosine to guanine. They always match up across the middle like that. And if we look carefully at them, what we'll realize is we said adenine, 1, 2, matches to thymine, 3. 1, 2, 3. But adenine to thymine, 3 rings across the middle. What about the others? Cytosine, 1 to guanine, 2, 3. 1, 2, 3. DNA always has 3 rings across the middle. And it's that rule that is determining what matches with what. If I have adenine, I must have thymine matching to it to have the 3 rings. If I have cytosine, I must have guanine on the other side to make it up to the 3 rings. And there are 1 or 2 other things we won't mention that actually go to determine this as well. But just trust me, that is the matching, C to G, A to T always. And in RNA, in actual fact, we'll see that the same kind of base pairing can occur, but it occurs within the molecule. It doesn't have another molecule running next to it. It occurs within the same RNA molecule, but I'll show you that more carefully in a minute. Go back to the little acrostic that I gave you before, and we said agree, A, G, agree, 2, 1, 2, purify, purines. They are called, 2 ring structures are called purines. Agree, 2, purify, 1, cute, C-U-T, pyramid. These single ring structures are called pyrimidines. So we always match a pyrimidine to a purine and a purine to a pyrimidine in constructing the DNA molecule. And we'll go, 1 or 2 other features of the DNA structure also become really important and result from this. I should just mention, this is something that was spotted very, very early on, before they ever knew the structure of DNA. A scientist called Chargas looked at DNA from many, many different species of animal and plant and everything. And he knew nothing about the structure of DNA at all. But he could analyze it, and they could analyze like the percentage of these different nucleotides. And he realized that whenever you measured the amount of cytosine, you always had the same amount of guanine. And whenever you measured the amount of thymine, you always had the same amount of adenine. And this is exactly why. These are called Chargas rules. All DNA obeys it. You always have as much cytosine, you have guanine. Why? Because they have to match. Whenever you have a cytosine, you have to have a guanine. It's 1 to 1. Whenever you have an adenine, you must have a thymine. A to T, it's 1 to 1. So no matter how much of the one, you have an equal amount of the other. Excuse me. Okay, so we also have learned that DNA is a double-stranded molecule. When this model was first worked out by Watson, Crick, Wilkins, et al., they struggled to put these molecules together, and until they had this amazing insight into the structure. Remember what I told you. We have here a 3'. This is the 3' hydroxyl. This is where the bases get added. And on the other side here, we have our 5' carbon. This means that our molecule here has an orientation, right? It has an orientation. There's a 3' here, 5' up, 3' down, 5' up there. And here they are joined, here the one strand, all joined together like this. But they struggled to see how it was that the T matched to A and G matched to C until they realized that in fact the molecules on the other side are turned upside down. So this here, look here, we've got a 3' end there, and we've got a 5' end there. And we will refer to these as being the 3' end and the 5' end. And this is running 3' to 5'. But the other strand is actually running in the opposite direction. And for various reasons, where they realized this was the only orientation in which these bases could actually join to one another. So on this strand here, we have the 3' end up here and the 5' end down here, 3', 5', 3', 5'. Whenever you look at a DNA molecule, whenever you look at the strand, or you're trying to decide what the other strand is, if you've got 5' up here, the other strand, that 3' must be there. Okay, 5' always matches to 3', 3' to 5'. The reason that they realized this was because they could work out how the adenine and thymine actually formed their bonds that keep them together. These bonds down the backbone here are extremely strong covalent bonds. But the molecule is held together by much weaker bonds that are these bonds here, which are hydrogen bonds. Very weak bonds, but remember there's a gazillion of them. These are very long molecules with many, many, many of these hydrogen bonds. So the DNA molecule is actually very, very stable. Under normal conditions in the cell, the DNA molecule is extremely stable because of how common these hydrogen bonds are and because of the strength of these bonds down the backbone. But see the backbones running in opposite directions, and that allows these hydrogen bonds to form. Just as a little aside, notice between thymine and adenine, there are 2 hydrogen bonds. Between cytosine and guanine, there are 3. The cytosine-guanine bond is much stronger than this one here. So running that way and that way through the whole molecule. The reason I'm emphasizing this so much is because this becomes very important in the next topic that we're going to discuss, which is how DNA actually doubles itself up. This becomes very significant. We say that these, by the way, And these are anti-parallel. They are parallel, but they run in opposite directions. So they are anti-parallel. And the whole thing together forms this beautiful lazy spiral in space. So just give me one sec. Sorry, I just had to blow my nose. Okay, so have a look here. If I've got a DNA strand, I've just got one side of my DNA strand, okay? Let me say here, oops, sorry. Let me say I've got this side here, right? And this side, for some mysterious reason, has disappeared, is gone. I can very easily construct the other strand, the matching strand, because I know that if I've got one strand here, the matching strand A must have a T, T must have A, G must have C, etc. So here we have a strand 5', A, T, T, A, G, C, C. What is the matching? In your minds, do it. First of all, always, what is the priming? If this is 5' on this side, what is this strand here? It must start with 3', okay? 5' to 3'. What's that matching? What does that match? What does that match? What does that match? That, and that. Yes. 3', A to T, T to A, T to A, A to T, G to C, C to G, C to G. This strand on the one side of the DNA, 5' ATTAGCC, must be matched on the other side by 3' TAATCGG. The priming gives you the directions of the molecules and tells you that they're running opposite to one another. Do another one. 3' GGCAGC GGG to T AAAAA. Correct. C C G T C G C A T T T T, and it's running 5' to 3', where this one is running 3' to 5'. Easy peasy. Okay. And just so this is the doubling up of the DNA molecule, and this is a typical kind of problem that would be set. Always remember, carefully examine this, okay? Carefully check the priming when you're answering. Now that is for DNA, that's just kind of interest, right? That we can do this, we can make up the strands because we know the one strand. It's actually the way in which the cell works. This is how the cell works. This gives a very quick and easy way for the cell to replicate its DNA. And remember that every time a cell divides, it has to replicate all of its DNA. And in actual fact, in reproduction in eukaryotes, they divide twice. And the doubling of DNA is a critical part of cell division of whatever sort you're talking, whether you're talking mitosis or meiosis or whatever. The replication of the DNA is critically important because all of the DNA has to be replicated. Each cell that results from the division has to have a complete copy of the DNA. It has to have all the requisite information, and it has to, if it's in mitosis especially, it has to be identical. It has to be absolutely identical. In bacterial cells, which only divide by binary fission, each bacterial cell has to have a complete complement of all of the information that the original mother cell had. Otherwise, they're not going to function properly. So the DNA has to be accurately replicated each time the cell divides. Not only that, when the DNA is being replicated, the genes which are busy being replicated cannot be used. They cannot be read off and used by the cell as long as division is taking place. So division has to take place as fast as possible, and so that you take those genes out of commission for as short a time as possible. So DNA replication has to be rapid, and it has to be extremely accurate. And the simple rules that we've applied here are the reasons that that takes place. And the reason that they can take place, first of all, each strand is a template, and it's a template for the production of the other strand. Go back here and realize, if I took away this strand here, you could recreate it from that information. Just the same way you created this strand here from that information. So each strand is a template for its for the partner strand. And the way in which the cell is going to replicate its DNA is fundamentally what it's going to do is it's going to split the two strands apart like that. And it's going to use them to recreate the matching strand on each side. It becomes a little bit more complicated, naturally. This is biology, okay, right? So it's the whole system is very carefully governed, and it is run by a complex set of enzymes. The very first enzyme that we're concerned with is this one here called DNA helicase. There it is there. And helicase is responsible for actually opening up the DNA strand. And when a cell sees single-stranded DNA, it has mechanisms which are designed to actually digest single-stranded DNA because it looks like a virus. So when a replication is taking place, the single-stranded DNA gets stabilized and protected by these proteins which are laid down, which are stabilizing proteins. And they maintain, for a little short distance, they maintain the freedom of the two strands so that they don't wrap around one another and they don't get damaged by the cell's protective mechanisms. So we've opened it up. This here, the division, this Y-shaped division here is called the replication fork. So there's the replication fork there. And realize that this helicase is running in this direction here. It's running in that direction and opening up the strand. So when we look at the geometry of what is happening, we're going to see that there's one strand here which can be distinguished from that strand there by what happens. And what happens is dependent on the direction of opening. So keep in mind, we're opening in that direction. So we got now on either side, we have single-stranded DNA. We understand that it's actually relatively simple to reconstruct by just bringing in matching bases. If we got AA there, we bring in TT. But there's a couple of steps that lead up to this. And the first one is one that you must remember. You must always bear in mind this first important step. When I am replicating DNA, I cannot do it straight away. I cannot just start producing DNA, a matching DNA strand. Instead, a short matching RNA strand must be laid down. And this is called the Rna Primer. It can happen very easily. This little area here is a hybrid between DNA and RNA. In other words, if I have a thymine on the DNA here, I would have a uracil matching it there. I'm sorry, if I had an adenine here, I would have a uracil matching it here, right? Otherwise, the matching would be the same. It would be C to G, C to G, C to G. If I have a thymine here, I'd have an adenine. But if I have an adenine here, I'd have a uracil. because this is RNA here, the little short piece of RNA, and that is called the RNA primer. And nothing will happen. No more replication will happen until this little primer has been laid down. The reason I emphasize this so much is we need to remember that to understand the rest of replication, but also bear it in mind when we talk about genetic engineering. We're going to hear about this RNA primer over and over and over again. It becomes a very useful little trick for us to use in genetic engineering. So in the natural system, there goes my RNA primer. Once the RNA primer has been laid down, and by the way, it's laid down by its own polymerase called RNA polymerase. Once that is laid down, then this other polymerase here, DNA polymerase, comes in and it starts laying down all the DNA bases from there on. Here's why I said it's so important to make note of the direction, because have a look now at the orientation of our strands. Here is the 3' end of this strand. Follow it all the way down, and we end up here. Here is its 5' end. So here is going 3' to 5', being opened up. Well, what did I tell you? Whenever we lay down a nucleic acid, whenever we polymerize a nucleic acid, we can only add at the 3' hydroxyl end. Now, this is a little bit difficult to visualize, but just imagine one base being attached here, okay? And let's say 1 or 2, okay? And we'll say, what is their orientation? Well, if this parent strand here is 3' to 5', the new strand orientation must be 5' to 3'. In other words, what have I got here at this base here, waiting to have another base added to it? Where is its reactive site? It's 3', right? It's 3' hydroxyl is there, 5' to 3'. We always grow strands in the 5' to 3' direction, because it's the only, at the end, it's the 3' hydroxyl, which is reactive. We're adding bases here. We're not adding them here. We're adding them here on the new strand. And they're adding always to the 3' hydroxyl, 3' hydroxyl, 3' hydroxyl, 3' hydroxyl. As the DNA polymerase advances, it lays down a longer and longer and longer strand behind it. And this is called the leading strand. It is a leading strand because it's going in the same direction as the helicase. Helicase is moving that direction. DNA polymerase is moving in the same direction. That's fine. For this strand, it's very easy to understand what is happening. But on the other strand, it gets a little bit more complicated. And its complication is because of this rule. We have to lay down primer. On the leading strand, it's fine. All we have to do is lay down primer once, and we can proceed to add tens of thousands of bases in a long strand thereafter. We don't have to worry about it again. But on this strand here is called the lagging strand. And it is the lagging strand because we are actually going to have to add bases in the opposite direction to the way the helicase is operating. Helicase is going this direction here, and the strand is being opened up, but look at its orientation. If that's the 3' there of the original parent DNA strand, here's its 5'. Okay, so now what are we going to do? Because we always have to add on our new strand, we have to add in this direction, right? So I have to add in that direction. This strand, the leading strand, is fine. I'll add in that direction. This strand, the lagging strand, has to be, we have to build our strand in the opposite direction. We've got a problem because before we can start adding DNA bases, we've got to lay down an RNA primer. which means we've got to open up this replication fork enough that I can lay down a primer, and I'm going to lay it down here, there, because I've got to add in that direction. So I lay down a primer here, there, and I'm going to start adding 5', 3' direction here, and bam, what I do, I smack up against a strand that has already been laid down. In the meantime, while I'm busy laying this down, the strand down here, the helicase is meanwhile opening up the strand, the replication fork further down here, and we've got more single-stranded DNA behind the strand we've just been busy making. What have we got to do? We've got to lay down a primer again, and now make another little short piece of DNA. Boom! It bangs up against our primer here. Bangs up against what we've laid down before. Goes on. This replication here takes place in a disjointed fashion. We end up making short pieces of RNA, DNA, RNA, DNA, RNA, DNA, and those are called Okazaki fragments. Here they are here. Here's the name. These are Okazaki fragments, and they arise because we've got to lay down our bases in this direction, away from the direction that we're opening. And we have to repeatedly lay down primer, make short piece of DNA, then stop. When we reach the old primer here, we've got to stop. In the end, we end up with a whole series of these Okazaki fragments. But Okazaki fragments consisting of a piece of DNA, piece of RNA, piece of DNA, piece of RNA. But fortunately, there is another kind of DNA polymerase, which will come in, and it can actually digest out all of the RNA, and it will replace the RNA with DNA. Same thing will happen here. It will replace this primer with DNA. And here, it will replace all of the RNA primers with DNA. And we then have this enzyme here, DNA ligase. Ligase, what it's doing, it is ligating. Ligation means to join. So DNA ligase now comes in, and it seals up any gaps that have occurred along. In fact, we have a little gap in between each fragment once all the DNA polymerase has eaten up all of the RNA. So there are some gaps there, and they are healed up by this DNA ligase. So when you want to understand what is happening, remember these things. First of all, we can only add at the 3' hydroxyl end of the growing strand. So it has to go 5', 3' direction. Second, when we open up the strand, one of the strands will be opened and will be replicated in the same direction as the opening. That is the leading strand there. The other strand is the lagging strand, and it has to be replicated in short, disjunct fragments called the Okazaki fragments, because we have to wait for this fork to open up enough to lay down an RNA primer and then lay down DNA, new DNA in this direction. And so that's something of a complication. I will tell you that in actual fact, in a cell, all of these ends, they don't like fly in separately and operate, then fly away again. They're all collected into this massive unit of joined enzymes called the Replication Factory. And this whole process, making this strand here and making the lagging strand, is done in an extremely efficient fashion, very, very rapidly, with minimal error. Some errors may creep in, but there's one feature of this which is tremendously important, and that is that this DNA polymerase here has proofreading ability. It will proofread the new strand, and it checks it against the old strand to make sure it's always brought in the correct base. If it hasn't, it will stop, go back, digest a few bases, and then repeat itself and put in the correct ones. So this proofreading ability of the DNA polymerase enormously increases the accuracy of this replication process. Okay, so yeah, I've given you here a summary of, please make sure that you study this carefully and that you really understand exactly what is happening. Okay, so here's a good summary diagram for you. Again, where we're opening up in this direction here. This is our leading strand here. Here's the lagging strand there. Here's the DNA polymerase running down the leading strand there, just making a long continuous strand. Opened up by Helicase at the replication fork. And then the primers are laid down. You will see this name here. It's primase. Primase is an RNA polymerase. It lays down an RNA strand, but we refer to it as primase because it's specifically laying down primers. The DNA is laid down by DNA polymerase, and there is another kind of DNA polymerase which will come in and replace the RNA with DNA to eat out all the primers and replace them with DNA. Any gaps are healed up by this enzyme here, DNA ligase. And so that is the basic mechanics of the DNA replication. Now, I'm not going to show it here because it always runs into problems with Zoom. But here is a little movie for you to watch just to go over the same steps. I will just mention that this process is an energetic process. It is an anabolic process. We are building a polymer, but we don't need additional ATP usage for the simple reason that these nucleotides, the free nucleotides, with their 3 attached groups, function much the same as ATP. They're basically exactly the same structure. We've got our base, sugar, and then we have these 3 phosphate groups here. So when I add to a new strand, what I'm going to, here I've got parent strand here has an adenine, and I'm trying to bring in, therefore, a thymine. I bring in a free thymine, and it has its 3 phosphate groups attached here. And in actual fact, what we're going to do is digest off 2 of these phosphates there. And in the same way as we've seen before, we're going to appropriate the energy from those phosphate bonds. And that is what is used to forge a bond between the phosphates and the sugar. So you get this phosphate group here is going to react with this sugar here, and it forms this strong sugar phosphate bond there. And here's our new thymine. The next one to come in, obviously, we've got a C there. We're going to bring in a guanosine with its 3 phosphates there. Guanosine triphosphate. Going to use the energy from the phosphate bonds, and we're going to join the G here. Remembering, these are covalent bonds. These bonds across the middle here are hydrogen bonds. So in eukaryotes, it's a little bit easier to understand how an entire DNA molecule, an entire chromosome gets replicated because they are linear molecules. A little bit more difficult to understand in bacteria because their chromosome is actually circular. So this is the circular chromosome here. In bacteria, there is an area of this chromosome which is actually attached to the cell membrane. And the replication of the DNA commences at that point. It's called the origin, or the ori. And the replication will commence from the ORI. But here's what you need to realize. This is true in eukaryotes as well. In eukaryotes, there are many origins of replication. Now, they're all scattered throughout the linear chromosome. There are areas where replication can begin. So if we go to the ORI, we're going to go to the DNA molecule and we're going to open up, a replication fork. But the thing is, when we do that, what we're going to do is we're actually opening up two replication forks, right? Because you're going to have one on one side, one on the other side. If I open up the DNA molecule like this, I've got one there and one there. So when we start this replication, we run it in two directions. And this is exactly what happens in the prokaryotes. The replication runs in that direction, and it runs in that direction there. We end up with two strands here. It runs around and around and around until they meet. And they're going to, those two replication forks are going to meet at the origin again. And in a circle, just because of the geometry of it, in the circular chromosome of the bacteria, we end up with these two chromosomes tied with one another like that. So what happens then is that an enzyme actually comes and it cuts the DNA. And then that same enzyme we heard about before, DNA ligase, just repairs this little cut in the DNA, and the two chromosomes separate from one another. In bacteria, those chromosomes usually are pulled apart by cell growth. They're tied to some proteins, and the cell grows and actually pulls them apart. So here's a beautiful electron micrograph of a replicating bacterial chromosome, and its replication is just about complete. So, but here's the replication fork. There's one replication fork there, and here's the other replication fork there. And they have run around. They've began somewhere over here, and they began somewhere and have run all the way around and have met here. And when they meet, these two circular chromosomes will be tied. The cell will cut them and separate them like this. In the eukaryotes, there are many of these origins of replication. And the reason for that is that there's so much more DNA. The chromosomes are enormously long in eukaryotes. And in order to effect replication efficiently, there have to be many origins of replication. The same thing applies in actual fact. As soon as we open up origin of replication, the middle of a chromosome, we've got two replication forks. All of the replication forks run together, and then the new strands separate. The other important thing to realize in this replication scheme is that when we look at our strands, at the final product, what we see is that each of the strands consists of an old strand there and of a new strand there. Same on the other one. Here's the original parent strand there and there. They've separated, and now they've got new strand. Because whenever we replicate the DNA, we always keep half of the old parent strand and lay down new strand. It is called semi-conservative. It is a semi-conservative replication.